afternoon, and thank you for joining us today. Welcome to our second installation of the Brain and Behavior Research Foundation webinar series. As many of you know, we are formally NARSAD, the National Alliance for Research on Schizophrenia and Depression. We were founded in 1987, and we are continuing to award NARSAD grants, having already awarded almost $300 million in grants to more than 3,300 scientists. Please check your mailboxes and your inboxes in the next few weeks. You will be receiving more information from us outlining the next in the Meet the Scientist series discussions. I'd like to, just before I introduce our speaker today, ask you to continue and enhance your absolutely wonderful support of the Brain and Behavior Research Foundation in order for us to continue to be able to do things like this, but more importantly, to support the scientists and the hope for all of our loved ones. It's a privilege to have Dr. Weinberger of the National Institute of Mental Health talk with us today. He has been a Brain and Behavior Research Foundation Scientific Council member since 1997 and a NARSAD grant recipient. He has identified brain mechanisms related to a number of genes found to cause psychosis. This work emphasizes how genes interact with each other and the environment. Today's topic is, how do genes cause mental illness, which is the focus of Dr. Weinberger's research. Dr. Weinberger?
Thank you, Benita. It's a great pleasure to be here with the Brain Behavior Research Foundation community, formerly the NARSAD community. I have done this before. I look very much forward to the opportunity to do this. NARSAD, the Brain Behavior Research Foundation, has been absolutely instrumental in backing and supporting research in understanding psychiatric illnesses. It is the American Cancer Society of Mental Illness Research, and most of us who do this work would not be able to persist at it were it not for your support. So, again, I'm very appreciative of having an opportunity to talk to you about some of this research. So what I thought I would do today is try to give you a little overview of some of the insights that are emerging from studying genes related to psychiatric illnesses. You don't have to be a psychiatrist or a neuroscientist or a genetics researcher to know, just by reading today's newspapers, that the capacity to study genes, which has emerged as a mainstream of science in the last 2 decades, has changed essentially all of biomedical research and the understanding that we have about most common human conditions, from cancer to diabetes to heart disease to mental illnesses. And psychiatric illnesses are being profoundly affected, impacted by our discovery of genes that seem to relate to who is at risk for them. So let me launch into this a little bit. I'm going to go through a bunch of slides. There's a few key points that I want to make in this presentation without going into too much detail. And I just want to give you a little overview of the key points that I'm going to make. The first key point is that the genes for mental illnesses, like schizophrenia, bipolar disorder, depression, etc., are not genes for these disorders. They're genes for about the function and development of the brain. The second point I'm going to make is that these genes are about very early things that happen during the development of the human brain. Those are the key points I want to make, but I will illustrate this with several examples. Let me just remind you on the next slide that everybody knows that there's something inborn about how people feel about the world around them, how they perceive their environment, how they respond to it, how they negotiate the vagaries, stresses, unexpected complexities of human development and life. And, you know, we know this in our families, that no two kids are identical, even if they're identical twins. They're not totally identical, but most of the variation within people in many basic personality characteristics, and including many basic aspects of how we think and feel, are related to genetic variation in the human population. And these variations we inherit from our parents. Let me remind you on the third slide of why we study genes. And this, I think, is really the essence of the issue. We know from a century of doing family studies, of doing twin studies, and of even doing studies of children adopted away at birth. trying to differentiate what is environmental from what is genetic in terms of risk and development of psychiatric disorders. We have learned that most of the risk for psychiatric illness in a population is related to inheritance. Genes, in contrast to everything that's been studied about mental illnesses in the last century, transcend phenomenological diagnosis. What I mean by that is, until very recently, until we had the capacity to study genes directly, we didn't have the technological capacity, we didn't have the sequence of the human genome, all these major breakthroughs in genomic science, which have allowed us to use genes as biological tools to understand human behavior and the brain. Before we had this information, most of research in psychiatry was comparing something about an ill person with something about somebody who wasn't ill. And we could do brain scanning, we could do cognitive function, we could do EEGs, we could measure things in the blood. But all of these studies are what are called phenomenological studies. They compare a characteristic of one sample, here a sample with illness, to a characteristic of another sample. In those kinds of studies, which are observational and phenomenological, just the phenomena associated with illness, it's impossible to know how central any of the phenomena are to the causes of illness. They could all be secondary manifestations of being ill, of medication, of the mental state of illness, of the mood state of illness, and many other secondary factors that occur to ill people, general medical health, for example. So without genes, there was no capacity to understand the basic mechanisms of causation. Genes, by definition, represent mechanisms of disease. This is a key point. It's not that they represent the only mechanisms of disease. They may not even be the most tractable from a therapeutic perspective mechanisms of disease. We know, for example, that the people who develop lung cancer, smokers, is very much about their genetics. But if you want to have a huge impact on lung cancer, the best way to reduce the risk of lung cancer is not through genetics, it's through public health programs that reduce smoking. So it's not necessarily to say that genes will be the answer to the new therapeutics of psychiatric illness, but they represent the first and only clues to the objective mechanisms of disease that we have ever had. And because of these insights, it is really safe to say we have learned more about the basic causes and mechanisms of mental illness in the last decade than we knew in all of past history. Now, another reason to study genes is that they clarify the environment. In fact, it's often said that one of the reasons to study genes is so that you can finally understand what the environment is, because people experience the environment on an individual level very differently. And the toolbox that people have to handle their environment, to negotiate the environment, this toolbox is the genes that we inherit from our parents. I like to say that if you think of life as kind of a monopoly game, genes will never determine how many railroads you own or whether you have a monopoly on Marvin Gardens or Park Place. But genes represent the only square on the board that you absolutely know with certainty, which is the go square. It is the toolbox that you play the game with. Genes also identify in an objective way at-risk individuals because these genes are the risk factors. And finally, probably most importantly, genes will identify biological pathways for the development of new treatments. Every medical treatment used in the treatment of psychiatry today, without exception, was discovered without any knowledge of the causes or pathogenesis of psychiatric illness. They were all discovered, for the most part, because of some accidental observation based on their utilization for some other purpose in medicine. We have to do better at finding new treatments now that we have some biological clues to the causes and mechanisms of illness. We're not going to change people's genes, but we're going to use genetic insights into how cells work to help us find ways to modify those effects. If you remember, genes are about two things. They're about fixed traits that we have all of our lives, like hair color and eye color, but they're also about the blueprint for the manufacture of proteins in a cell from moment to moment guide the biology of those cells. Now, there's one other point I want to make about psychiatric genetics, which is on the next slide, which is a key sort of fundamental point. Psychiatric genetics is like the genetics of most common medical disorders. It's the same as the genetics for diabetes, heart disease, high blood pressure, stroke, cancer in many ways. These are diseases that are called polygenic or multifactorial and genetically heterogeneous. Let me illustrate what that means by looking at this slide. If you look at these brown circles have an illness. These are representatives of people, and they have an illness. Let's call it the Brown trait. It's a trait, meaning it's a manifestation that's genetically determined to some degree. Each of the people that have the Brown trait have several puzzle pieces that represent the risk factors or causative factors for them having the Brown trait. You notice that nobody has only one puzzle piece that has the Brown trait. Everybody has multiple factors that account for them having this Brown trait. This is what's called polygenic. There are multiple genes that together combine, fit together just in the right way to account for the risk profile of these individuals. It's like saying, to get a stroke, you need high blood pressure, you need high cholesterol, you need other problems. You fit multiple factors together into an architecture that determines the overall likelihood or liability that you will become ill. The other thing to notice is, in addition to the Brown trait being polygenic, every person with the Brown trait does not have the exact same combination of risk factors. Different combinations can join together to produce the same, what's called phenotype or clinical manifestation of the genetic risk architecture. So this is the notion of heterogeneity. Every person that has the same manifest condition does not have exactly the same causative components to their risk bias or the risk liability. The last point to make is that there are a couple of circles, at least one, that has all the risk factors that anybody that has illness has, but does not manifest illness. This is because the genes are not enough. There have to be environmental factors, or there might be protective factors, and these are called non-penetrant cases. So this is the concept of genetic complexity. It's not as simple. These genes are not genes for your fate. They don't determine your fate. They determine your risk. Just like high blood pressure is not a determinant of stroke, it's a risk factor. Just like high cholesterol does not guarantee you a heart attack, but it does increase your risk. It is thought that these genes increase risk. On the next page, there's a list of a bunch of genes that have already been identified as likely candidate risk factors, true biological causative factors for schizophrenia. This is one of a number of candidate gene lists. I don't want to go into the details of the list, but I want to make 3 points from this. The first is there are variable levels of support for any one of these genes being a risk factor for schizophrenia. And it's likely that some of these will turn out to be false leads, but it's also likely that a number of these will be true identifying factors that determine somebody's likelihood in their lifetime to manifest these conditions. The second thing is most of these genes, which come by these weird acronyms that are large letters put together, and it doesn't really even matter what they stand for. Most of these genes, none of us even heard of 5-6 years ago, which makes the point, in a way, that the human genome did not evolve to validate some senior professor or famous researcher's pet hypothesis of what these illnesses are. The genes tell us what these illnesses are at a very basic cellular level, and that's the insight that the genes provide. The last point is that while this slide says schizophrenia susceptibility genes, none of these genes have been found only in association with schizophrenia. In fact, most of these genes have also been found for bipolar disorder or for autism or for attention deficit hyperactivity disorder. And this makes a key point also. The human genome did not evolve to validate the DSM-4 criteria of psychiatric diagnoses. These diagnostic criteria are arbitrary. They're determined by a consensus of clinicians based on experience and phenomena. They are not based on any biological understanding of the origins of these disorders or of their basic mechanisms. These genes tell us what these disorders are at a very basic cellular level, just like genes for cancer tell us what cancer is at a very basic cellular level. But the genes also make the point that these diagnostic categories are artificial and imprecise. Now, I want to make a point in this cartoon slide that I've presented several genes and I've presented a perspective on how to think about them. But there is a great deal of controversy in this field. And there's a great deal of controversy in behavioral genetics for many decades because behavioral genetics, by definition, can be misused, it can be misinterpreted, it can be both overinterpreted and underinterpreted. It has a checkered history of trying to explain behavior by genetics, and I think we have to respect that, but also recognize that the science requires objective, rigorous experimentation to understand the role of genes and to put them in the appropriate perspective. Genes will not, as I say, determine whether you own a railroad monopoly or other aspects of your life, but they do bias the probabilities. So this is the field currently is in a somewhat a state of sort of division about whether or not we have good evidence for genetic associations with psychiatric illness or not. And there's pretty much two schools of thought these days. There's a school of thought that only sees the inconsistencies in the research literature. There's another school of thought that looks at the wine glass, sees wine in it, and says it's not half empty, there's plenty of wine, and the wine has to do with the fact that, as I mentioned earlier, these are very complicated medical states with many factors that contribute to the manifest condition and tremendous heterogeneity. Every person around the world that we give our same arbitrary, imprecise diagnosis to does not have the same underlying biological disorder, so we would expect all these consistencies. But this is just to give you a flavor for where this field is right now. So let me get to two questions to move on to the next point of this talk. Why are genes for psychiatric disorders so controversial, and why is it so complicated? And there's one fundamental answer to both of these questions, which is that, as I said earlier, the genes for psychiatric disorders are not for psychiatric disorders. And so the question is the wrong question to begin with. Genes do not encode for psychiatric phenomena. Genes do not encode, and genes represent the code for a cell doing something. Genes do not encode for hallucinations or delusions or panic attacks or manic episodes. Genes don't do that. What do genes do? Well, this is a diagram to make the point that genes encode simple molecules, mostly proteins, in cells. That's all they do. Genes do not know from hallucinations or delusions or panic attacks or manic episodes. They know nothing about human behavior. They only know about the regulation of the biology of a cell. Now, what does that mean to a human being? If you inherit a variation in a gene, which means that the sequence of the letters of the DNA code has changes in those letters, the wording is a little different. If you inherit those variations, this will affect the function of some particular aspects of a cell, of cells in your brain. And a way of thinking about it is if you inherit a variation in the genetic sequence that affects the function of the DNA of a particular gene, it will produce a molecular bottleneck of the function of a cell. That's it. That's all the gene does. How does that impact on behavior? What that does is that leads the cell to respond abnormally or slightly in a modified way as it grows up from the first level of fertilization in an embryo to the growing, developing fetus and the growing, developing human. As the brain develops, cells interact with other cells to form synapses, connections, networks. As the brain develops, those cells and connections and networks become circuits in the brain, systems in the brain. Cells process molecular information. Brain circuits and systems process environmental information, which is sensory information, emotional information, and cognitive information. The clinical manifestations of psychiatric illness are the what we call emergent phenomena, the most complicated phenomenology of the processing characteristics of these circuits and systems, which evolved, developed, and grew up based on the molecular biology of the cells, which were instructed to behave as cells by the DNA that you inherit from your parents. The more we can study genes related to mental illness at the level of brains and cells, the better we can understand the mechanisms of how they translate into illness, and the better we can use them to find new biological clues to understand better approaches to treatment. Let me put this in a totally different frame of reference just to make it more logical. Because when you think about listening to music from an orchestra, there are many components to how the music has to ultimately sound like a recognizable form of music. So the genes represent the score, the cells represent the instruments, the individual instruments, the distributed neural systems are the woodwinds and the brass and the strings, and ultimately all of this has to work in some very representative way for us to recognize it as music. Our assumption is that there are slight changes in the score which affect the individual behavior of the musicians in the orchestra that disrupts the overall the combination and synchrony of the different sections of the orchestra to make this sound like something other than the music that we expect it to sound like. I want to make a point about the fact that if genes are about the brain development and function, we should be able to observe evidence of this in people who have the genes but don't have psychiatric illness. And there have been many studies of this over the last 20 years. We actually started this work in my lab in the early 90s, where we studied identical twins who were discordant for schizophrenia. These were people who were genetically identical. They have the same families, the same environment. They share essentially all their genes and essentially most of their environment, but they don't share illness. One twin is ill, one twin is well. And we asked the question, what was the same about the identical twins when it's clear that illness was not the same? And one of the things we noted that was the same or more alike than it should have been, given that they were not both ill, was apparent cognitive problems. That even the well twin had some of the cognitive problems that patients with schizophrenia have, arguing that what was genetic was not really the illness diagnosis, but something about the function of the brain that could be observed by doing cognitive testing. And this is results from a recent paper that was published last year in a very large family and twin study of over 2,000 subjects. And the take-home message here, which is shown in the red, is that the genetics of risk for illness and the genetics of cognitive function in these families basically are the same genes, arguing that what the genes are about is not our diagnoses, but the subtle aspects of how you put a brain together from early in development, which have a number of ways they can show that the overall wiring diagram had some slight perturbations in it. And one of the ways they show that is by variation in cognitive function. In addition to studying cognition, that is how well you perform on tests of memory or intellectual function, we can also study how the brain actually performs those tests, how it actually handles cognitive information. And this is going back to the brain in my slide that went from a gene to a cell to a brain, because the closer we get to the biology of these genes, the more these effects should be obvious. And so we can study the brain directly, not at the level of behavior at all, using these modern neuroimaging techniques. And this is an example of how that's done. This is a study using a technique called functional magnetic resonance imaging, which does not use any radioactivity. It's totally harmless, totally painless. Person lies in an MRI scanner and does a simple cognitive task. And this is a task called the NBAC working memory task, which is a task about how to use your past experience to guide your behavior in solving a simple problem. It's something that patients with schizophrenia have disproportionate difficulty with. It's something that their healthy siblings have some difficulty with. But what we show in these data, in these representations of the brain, is that both patients with schizophrenia and their healthy siblings who have no psychiatric illness whatsoever share a characteristic of brain function related to doing this kind of cognitive task, which we call inefficient or untuned or less tuned. It's like they have to work a little bit harder in their brain to get the same level of performance. The way we like to think about this is they can drive from one place to another, they get there on time, they get there in perfectly good shape, but their brain gets less gas mileage in the process. And we think this has something to do with the basic biology of how these circuits got put together, that when they have to handle this kind of complex information, the overall architecture of the circuitry, based on the genetics of risk, is such that the circuitry is a little bit less efficient in managing all this. Now, I made a point repeatedly that the genes are not enough. Genes exist as a risk architecture, but they don't determine your fate. They determine how much at risk you are. Other things have to happen to you to move from being at risk to being ill. Now, and this is the whole concept that genes interact with the environment, and they interact with the environment to exaggerate the effects of both. So if a gene interacts with the environment in such a way that the environment adds risk to the likelihood of being ill, it will exaggerate the gene effect. And the gene, by definition, because it makes you more sensitive to certain environmental events, will exaggerate the environmental effect. The exact same environmental effect in somebody who doesn't have a risk gene that would be affected by it does not show an effect. This is the nature of interactions. So here was a study that we did several years ago looking at another risk factor that's been associated with risk for schizophrenia called obstetric complications. It's been known for a number of decades that in the histories of many individuals with schizophrenia in adult life, not everybody, but some, there was a history of difficult gestation, which could be noted because there was hypoxia at birth, there was a blue child, there were poor initial responses at birth, etc. And we looked at a bunch of genes that have been associated with risk for schizophrenia in families where there was a history that the offspring who developed schizophrenia had a complicated delivery or birth. And it was complicated delivery associated with what's called anoxia-ischemia, which is too little oxygen. And what you can see here, if you just look at the column that says OR, which is the odds ratio, and this says how much of a difference did it make in the likelihood that these people would have schizophrenia as adults if they inherited both a form of the gene and had present obstetric complications. And there's one gene in particular I want to draw your attention to, which is this gene called BDNF, which is a very important gene in early organ development, very important in early brain development, but in other developments, other organs also. What you see here is, if you inherit this gene in a particular form, and you have an obstetric complication, the likelihood that you would have schizophrenia compared to people who had neither of those two risk factors was increased 12-fold. This is a very substantial increase in risk, and it illustrates how when you put multiple risk factors together, they can have much more of an impact. What made this very interesting to us was that this particular change in the BDNF sequence did not affect BDNF in the fetal brain. It didn't affect BDNF in the mother's brain. It affected the function of BDNF in the placenta, which raises another very interesting issue about there are many factors, both within the fetus and external to the fetus, that contribute to the environment of healthy brain development during uterine development. And this is another clue to the complexity of the factors that contribute to disrupting brain development in individuals who ultimately will manifest schizophrenia during life. And this is my last slide to make this point about brain development. This is a slide looking at a gene that we've been very interested in as a risk factor for schizophrenia. It's a gene that regulates the entry and exit of a particular ion, potassium, into cells. Potassium is a critical ion for allowing cells to fire, making them, the cells fire impulses, and potassium is critical in their firing impulses. And we've shown that this gene affects the firing of cells related to schizophrenia. And the interesting, remarkable thing here is the form of the gene that's associated with schizophrenia is this form called 3.1. And that's the bottom section of this slide. The top section of the slide is a form of the same gene that's not related to schizophrenia. What you're looking at here is the expression of this gene in cells in the human brain, actually in the frontal cortex, across the human lifespan. And we're starting at 14 weeks of gestational age. So on the x-axis, on the horizontal axis is age. This starts at 14 weeks in a human fetus and goes beyond age 80. Every dot on this slide represents a different human being's post-mortem brain. And this is work that we're doing here at the in my program at the NIH in Joel Kleinman's section. And what this demonstrates is the form of the gene that's associated with schizophrenia shows a dramatic increase in the expression of this gene during fetal life. It is a much more active partner in the development of the brain than it is in the workings of the adult brain. This is the opposite of the gene that was not associated with schizophrenia. And we have seen this pattern of variable expression of the genetic factors linked to schizophrenia being much more critical for the activity and function and biology of the fetal brain than the adult brain, which again makes the point that the genetic risk factors, just like obstetric complications and probably many other risk factors, for the adult emergence of a condition like schizophrenia is very critically dependent on what's happening early in the development of the human brain. So I've tried to tell you a few what I think are simple truths. Let me just remind you of some of the points I've tried to make, and then I'm looking forward to your questions. Genes tell us what psychosis is at a basic cellular level. The current evidence converges on subtle molecular bottlenecks in diverse aspects of synaptic processing and brain development. Synaptic processing means how cells communicate with each other. There are many pathways to what we call schizophrenia. This is true of autism. This is true of bipolar disorder. This is true of cancer. This is true of heart disease. These are not singular disorders with one specific way to get there. Many, many roads lead to this particular outcome. Genetic risk for schizophrenia and for cognitive deficits in affected families share most of their genetic components, arguing that these genes are about much more fundamental aspects of how the brain is put together. The neural mechanisms association for many so-called schizophrenia genes involves tuning of this working memory circuitry. So this is a way of thinking about the genes at the level of how the brain works, not just at how the cells function. The molecular mechanism of association of many of these genes involves altered processing of fetally abundant transcripts. This seems to be the arena at which these genes are having most of their effects. And I think the take-home message is that simple models don't work. So that's it for my formal presentation. I would be very interested in your comments, thoughts, and questions.
Thank you, Dr. Weinberger. We really do have a lot of questions, and probably predictably a lot relate to some of the things that you've already talked about, risk and testing and the age of manifestation related to those risks. One of the last questions to come in said if we are hardwired at birth, then why is the age of manifestation often so late in life? But also, there were many questions regarding risk related to trauma, as you've already stated, obstetrics complications, including premature birth, dietary risk, stress was a recurring theme in the questions, and marital discord.
Right. Well, let me try to speak to a few of these things. I mean, so we know that early life experience impacts on brain development. This is very clear. And we know that genes are there from day one, and they dictate the developmental programs that life experience interacts with. That's the complexity of human brain development. It has to be that complex. If it was not that complex, human beings would not have evolved and survived for the millennia, many, many millennia that we survived, because there have to be enormous buffering capacities in complex programs of brain development, if there were not these enormously complicated buffering processes, we would be just too vulnerable to the vagaries of, starvation, wars, etc., the things that have, infections that have characterized human existence through the millennia. We would not have survived if we didn't have enormous buffering capacity to be able to withstand many of the things that happen in human brain development. That doesn't mean they don't affect us, but we have enormous capacity to find avenues of compensation. We know that the genes set the stage for how many of these things operate. Can you remind me what the first question? Oh, about late onset. So late onset is always a critical question. Let me make two comments about that. First of all, there have been many epidemiological studies, meaning large population-based studies, of the early life experience of individuals who will manifest psychiatric illness later in life. This is particularly true of schizophrenia. It's less true of bipolar disorder. But in general, what the data with schizophrenia is that as a population, and this is a little bit hard to get your hands around because at an individual case level, there are tons of exceptions to all the population-based rules. But at the population-based level, people who are destined to have a schizophrenic illness as young adults do show signs of early developmental deviations from, strictly speaking, the mean of the population. And and some of these things are very, very hardwired neurological developmental milestones. So, for example, the age at which you sit up, there are several large epidemiological studies saying that as a population, individuals destined to manifest schizophrenia as adults sit up about a month later as a mean, as an average. Now, obviously, most people that sit up at 7 months do not develop schizophrenia, and that's not by itself something that anybody would be alarmed at because one standard deviation of the population, a third of the human population, sit up at 7 months. But as a class, individuals who manifest schizophrenia as adults show that as their own population. The same is true with when you walk, when you acquire language. The history of enuresis, childhood bedwetting, is about 3 times greater in individuals who manifest schizophrenia as adults. All of these signs illustrate that there are manifestations of delayed maturation or some hiccups in the maturational programs very early in life, none of which would alert a parent by themselves to think anything other than, well, my child may have a little slowness here. Everybody has something growing up. So it's not anything that would be by itself sufficient to alert either a clinician or a parent. But it does, at the population level, identify that there's something associated with the later diagnosis that can be observed at the population level earlier in life. Why does it take 20 years for the psychosis to develop or the more serious manifestations? We don't have an answer to that, but the current thinking emphasizes that these programs that are slightly off track in development converge on the development of very highly complicated functional brain systems, particularly around the frontal lobe. And the frontal lobe of the human brain is a very late developing system because it's meant to handle very complicated environmental circumstances about independent living. And it's meant to handle conflicting circumstances, hormonally charged circumstances. It is the most highly evolved, most developmentally prolonged part of the human brain. And the current hypothesis that I think is most appealing is that these neural system deficits from very early in life converge on the function of this late functioning brain system, which when it comes completely online, which is early adulthood, it now can't function as an adult frontal lobe, and it manifests its failure in the syndrome that we call schizophrenia. That's sort of a thinking about that. Let me take another question.
Okay. Thank you. Very comprehensive. On the other side, of course, there are questions regarding treatments in the pipeline.
Right.
Repair treatments to alter genes,
right.
Any environmental protective factors, and additionally, should we be focusing on prevention in any way?
Okay. So those are really important questions. Firstly, let me say, because there was also an earlier question, if all of this is developmental, this is a very logical conclusion. If everything's developmental, it's all about the wiring, why would we think we can do anything about it? It's done. Why would we expect to be able to have an impact? So I think there are a couple of ways to think about that, and they converge on this question of new treatments. First of all, we had this old view that the brain is sort of hardwired, it's fixed, you can't do anything about it. We now know that that's an oversimplification. The same molecular soup that builds the brain maintains the brain. And we actually have been able to show in animal models, a number of people have shown this now, that some thought to be fixed developmental syndromes in animal models can be reversed. Even conditions as seemingly fixed as some mental deficiency states, mental retardation states, can be reversed in experimental animal models. Whether that happens in human beings, there's been a lot of interest in this possibility because the wiring is not as fixed as we previously thought it was. So there's hope that what looked like an immutable change may not be so immutable. Second point is that even if there are things that can't be undone or reversed, by understanding the molecular pathways that these genes operate in, the molecular systems in the cells, we can compensate for something. We might be able to put something in place that would block the effect of the fixed problem. I sometimes think about the gene networks as like domino games, and you can have multiple streams of dominoes converging at the end on one domino. You might be able to block many of the convergent streams by putting, a blocker in one of these streams so that it never gets all the way downstream. So that would be a way to use the genes to try to find new pathways. And then the other thing is, I think that, the expectation is that the genes are providing totally new insights. As I mentioned, most of these genes none of us even heard of 5 years ago. So they're opening up entirely new vistas for thinking about what matters to a cell that ultimately grows up to be a brain that manifests these psychiatric disorders. And the notion is, and this is true, it's being done in cancer, it's being done in all these other areas of medicine, this is the hope that we will have insights from these novel clues about the basic mechanisms to find targets for totally new approaches to therapy, totally new approaches that really get at the core problem that seems to set these dominoes going down the stream of dominoes. That's the hope of this. And I also think, one of the questions that always comes up about genetics is, does this create a whole new level of stigma because it sort of brands somebody as being at risk? My feeling is that this actually is another strategy, it's like all of scientific research and discovery, for reducing stigma, because it puts, again, these conditions back into the mainstream of medical science, where they clearly belong, just as, the elucidation of the molecular biology of Alzheimer's disease translated senility, which had plenty of stigma associated with it, into a now, general mainstream biological brain condition. And I think the insights that we get from genes and from these new molecular strategies for elucidating mechanisms, changes the landscape of how we think about these conditions.
I think we might have time for just a couple of more if I can speed this up. These are very personal to some of our listeners. One writer has a daughter, age 25, and she was diagnosed, at 25. She's now 41, and she is on medication. She lives at home with her parents, and they monitor her meds, but she hears voices constantly and verbally replies to them. The parent is wondering if that will ever stop. Is there any medication that is available to suppress the voices? And her constant verbalizing keeps her from working or volunteering. Would shock therapy help her, and how safe is it?
Right. Well, that's a complicated question. Firstly, obviously, this is a very difficult problem. I have a couple of thoughts that might be worth considering. First of all, every case is different, not just at the basic biological level, but certainly at the clinical level. And every individual who is ill, requires and will benefit from, a good working relationship with a good practitioner, psychiatrist in particular, with given the medication history here. And there's no substitute for that. I mean, that is obviously the first most important determinant of outcome. I understand that in many settings this is much more easily said than done, but let me just, since my job here is mainly just to provide what I think is the best vision for how things should get done, what happens in the real world obviously doesn't often make it possible to follow that vision, but that's the first thing. Now, in general, the medicine that overall has been most effective in reducing these chronic hallucinatory states is clozapine. I don't know if she's been on clozapine, but if she has or hasn't, obviously that's something very important to consider. Now, clozapine has its own collection of side effects, most notably the metabolic side effects and weight gain, and those are very serious side effects, but my attitude is always to try to get the best symptomatic improvement you can and then worry about the side effects as the next stage of intervention. The bottom line with ECT, with shock therapy for psychosis, is that it may work very transiently. It could reduce the voices, but the history and the experience with shock therapy for psychosis is it does not have lasting effects, and you end up chasing your tail, and it probably is not really advisable. So I think, I would argue that if she's still as symptomatic as she is, it would make sense to sit down and try to think about alternative treatment strategies, maybe some other medication to try to improve outcome.
I have one additional that, again, is a personal question. My son has lived with schizophrenia for almost 20 years, aged 19 to 38, takes multiple medications, struggles with memory and concentration. He forgets to wash his hair, shower. Sometimes the episodes last for 2 hours. What can I do to help him?
Well, again, that's a very tough problem, and I certainly understand how exasperating this sort of thing can be. I have a couple of comments, but it's a little bit the comment I made to the previous question. I think the most important thing you can do is have confidence that you have a psychiatrist involved who is knowledgeable, helpful, experienced, and you have a constructive working relationship with him or her to try to tackle this problem. In my experience, when patients are on these many medications, it's doubtful that they are all helping. Now, again, this is one person's experience, and it may be that every one of these medications does something beneficial to your son, but my personal guess is that they're not all useful, and they can contribute to some of the confusion and memory problems. In particular, I can't imagine that based on the treatment with Abilify and clozapine that there's any indication for benztropine, and that may contribute to memory and concentration problems. I would sit down and carefully review these medications and the reason that there's on so many of them. If there are truly these episodes of confusion, you need to rule out that this is not some, episodic neurological state, like some transient electrical seizure problem. It doesn't manifest as convulsions, and that's not inconceivable because these medications, particularly clozapine and dazipramine, could increase the possibility of some occult seizures. So again, this requires a careful review with a physician that you have confidence in on the treatment plan and the result of this medication strategy. I hope that helps.
Dr. Weinberger, thank you so very, very much. Your research and leadership in the field has been extraordinary, and it certainly continues to bring hope to so many people internationally, not just in America. As we know, federal funding is now on the decline for support of basic research, and our role is to step it up and fill that gap, keeping the field robust. So we certainly hope that those of you who are on the line will continue to support the work of scientists like Dr. Weinberger and make your donation today. Let me tell you that the audio and slides will be on our website as soon as we can convert it and get them there. and you will receive a thank you note from us with an opportunity to make a donation, of course. But thank you so much, all of you, for joining us. We know that this has been valuable. There were many questions that were not answered. We will attempt to work with Dr. Weinberger to see if we can follow up with you. So thank you again very, very much.
And thank you very much, everybody out there, for your support of mental illness research.